Welcome to our third day of the GEO Open Science Conference and All Hands meeting. And today we invited two people to give talks related to tonight's semi-final of Portugal against Wales. But the Wales person has not showed up. How did we know that Portugal would make it to the semi-finals? Well, we just knew it. But so, it's a great pleasure for me to introduce Maria Dornelas. She graduated from the University of Lisbon, and then she went to work on marine ecology of coral reefs at James Cook University with a colleague of mine, Sean Connolly, that was the senior graduate student in my lab when I went to graduate school at Stanford University. So that's the first time, let's say, that my path and Maria's crossed, through this indirect connection. And since then we have crossed a few more times as Maria's work has become really some of the best work done in looking at global biodiversity change, and she has become more and more interested in global patterns of biodiversity change. And so today is a great honor to have you here. Maria, welcome to the GEO conference.
So, good morning, everyone. Thank you for showing up quite early on Wednesday, peak conference fatigue day. I want to start by thanking Henrik for inviting me to be here. He's responsible for me being here in more than one way, as he explained. My PhD supervisor would not have accepted me had he not said I was okay. And I also want to thank the conference organizers for doing an amazing job. Okay, so I want to talk about what assemblage time series can tell us about biodiversity change in the Anthropocene. But before we really get started, I want to start by acknowledging a number of people who are well, I don't want to say partly responsible for the work I present here, in case I misrepresented, but they're definitely, they pull a lot of the weight for the work that I'm going to present. So first and foremost, Anne McGurran, she's the one who got me interested in Biodiversity Time series. She's the one who convinced me to give up a job in my home country to move to Scotland and join her in her ERC project, Biotime. But also Brian McGill, who's here, and Nick Gattelli, who have been involved in this work for a while and who have been amazing collaborators, really stretching what we can do. Also, Anne and mine joined a lab group, the Biodiversity and Behavior Group, and specifically Hideyasu Shimadzu, Faye Moyes, Kai Sievers, Laura Anton, and Miguel Barbosa there. And then I want to thank the S-Change working group. Some of us are here, Amanda Bates, Leticia, Diana, and Martin, and who have really, again, sort of pushed what we can do with Biotime data. And also, they're not here, but they co-led S-Change with me, Mary O'Connor and Sarah Supp. And then I also need to thank all the people who paid for this. So the ERC, the MASS, the Marine Alliance for Science and Technology in Scotland, the University of St Andrews, and IDEV. Okay, so let's really get started. So we live in a crowded planet. I believe this year there's 7.4 billion people living on this planet. The human population is growing by about 80 million every year, and it is set to continue to be growing in the foreseeable future. So it's gonna get more crowded. It's not less crowded. In addition to that, our per capita demands of resources on the planet are also growing. So not only there's more of us, we need more of the planet. And so that has led a number of people to call the times that we are living the Anthropocene. So a geologic era where humans have become one, if not the major driving force of of planetary processes. And we've done things like, for example, double the amount of nitrogen that's biologically available on a planet. We capture annually about half of primary productivity. And we burn so much fossil fuels that we are changing the climate. So it's justifiable to be worried that in our growing numbers, in our growing demands of the planet, we are really only leaving out the cracks of what we're not using for the rest of the biosphere. And so I think one of the biggest challenges of our time, like one of the biggest societal challenges of our time, is understanding how the Anthropocene is changing biodiversity. And it's a political challenge, it's a societal challenge, but it is also a scientific challenge, like it's a scientific question. And I'm a scientist, so that's how I look at this question. And so to answer these questions from a scientific point of view, we know how to do this. We need global, stratified, random sampling of biodiversity of the planet. This is what, ideally, what we should have. Unfortunately, the change part that's in this question adds a time axis. And I say unfortunately because I think that this is achievable. So the global stratified random sampling part of this is achievable. What is probably tricky is the time travel part. And unfortunately, our ancestors didn't really think to establish this global stratified random sampling of biodiversity on the planet. So we will have to also come up with ways to gather as much information about past change as we possibly can. And that's kind of what we set out to do within Biotime. So we set out to assemble as much a collection of assemblage time series that were publicly available. So before we actually set out, we sat down and came up with a set of criteria. We wanted to get as much data as we could. So these are the criteria that we came up with to be as inclusive as we possibly could. Right, so first of all, we demanded a minimum of 3 years of sampling, not necessarily consecutive. We're actually relaxing this criterion to just 2 years now because we're at the moment interested in maximizing spatial coverage and spatial representability in our data, but I'll get to that towards the end of my talk. So the reason for this is if you haven't gone there more than once, it's not a time series. To really estimate a slope, 3 points is the bare minimum that we need. We can estimate change with 2 points. Our second criterion was that the sampling method needed to be consistent through time, and this is because diversity is incredibly sensitive to sampling effort. So we need to be able to standardize for sampling effort, and the easiest way to do this is to have at least the way in which we collect samples to be standard, and then we can subsample so that we have a constant number of samples through time. Okay, and this is really, really important. Otherwise, we end up with biased estimates of diversity. And then the third and final criterion is that we were interested in assemblage level data. And what this means is that we go out and sample, and every species that we encounter in our sample we estimate its abundance. And there's different ways in which we can estimate abundances, and that varies according to taxon. So we're okay with that. We don't want just population data. We want everything to be reported. And the main reason for this, well, there's multiple reasons. One is we believe it provides possibly a less biased estimate for population trends. But the main and most important, the real reason that we started out with this criterion is that we were interested in quantifying change in alpha and beta diversity. So both Anne and I are really macroecologists at heart, and this is what we do. So this is what we set out to do. Okay, so it turns out that very quickly we realized that there's a huge amount of data out there. We started out with a conversation between Anne and I, and very quickly we realized we needed to hire someone to do this, and there's so much data that is available. That's not the limiting factor. And at some point we had to stop and actually start doing something with the data. We thought 100 data sets was a good place to do that. So our first paper that came out, that's what we had. By that point we had about over 6 million species abundance records. Over 35,000 species, spanning with time series spanning from the 1900s to the present. Although the vast majority of our data comes from the last 50 to 40 years. Okay, so at this point, I want to acknowledge two huge giants that were responsible for us finding a lot of this data. So OBIS and GBIF, these are the giants on whose shoulders we've been standing, and just sort of really, there's so much we can do because people have been mobilizing and creating a culture of data sharing and data standards that makes our lives as data users much easier. And so what did we find? This is what we found, and you may recognize this plot. I believe Enrique showed it on the first day, and Andy Purvis also showed it yesterday. It's the maybe infamous plot of no net change in species richness through time. So, but I'm going to maybe walk you through it and hopefully show you what I think this plot shows. So we have on the x-axis time, on the y-axis species richness. The gray points are the data, and we're really showing two models here. So the black line is what everyone pays attention to. It's a model that has a constant slope and different intercepts for each of the time series, and the slope here is really indistinguishable from zero. But there's also another 100 lines in this plot, and these are lines that come out of a model where we allow the slopes to be different for each of the time series. And again, like the really controversial line here is the black line. What I would like to emphasize is that really what we have here is a huge variety in change in species richness. So, and what I think this plot really shows is that maybe we should stop paying as much attention to the mean and start paying more attention to the variance here. And I guess specifically at this point, I want to point out to a paper that came out a couple of weeks ago in Nature by Josh Sinner and colleagues, where they do exactly that. And they look at the outliers, and they specifically focus on the outliers that are doing well. They're doing better than we would expect for the place where they are. They call these the bright spots. And they ask the question, what are we doing right in these places that recover from disturbance and that, and where humans manage to coexist with the rest of the biosphere? And so I think that's a really important way forward in which we need to move with this debate. Okay, so these results were somewhat controversial, but they have since been shown to be fairly robust. So they're consistent with a number of independent meta-analysis. In parallel with our work, Mark Welland and colleagues were assembling plant data sets, and they showed exactly the same pattern. In a different approach, Sarah Suppa and Morgan Ernest were looking at recovery, effects of disturbance on communities, and again, they showed a similar pattern, like lots of compositional change, but really not a clear signal in terms of the effects of disturbance on species richness. And more recently, Robin Elahi and Mary O'Connor and a number of other people have looked at marine data sets and came up with results that are entirely consistent with ours. However, I believe the expression in English for only looking at lines of evidence that are consistent with our worldview is cherry picking, and I don't want to do that. So let me also point out to a couple of lines of evidence that are, at least apparently, contradictory to our results. So the first one comes out of the Predicts work, and explicitly Tim Newbold's paper in 2015 in Nature, where they used a space for time comparison, and they estimate a loss of species richness in the terrestrial realm of 8.1% of species richness. Okay, so that's one line of evidence. The second line of evidence that's also in apparent contradiction with ours is this idea of a mass extinction. So a mass extinction is losing 75% of the planet's species. That's a pretty big number. How is that compatible with no net change in species richness? And we're talking about extinction rates up to orders of magnitude higher than background rates. So how can this be compatible with our results? And in a paper that Brian led and that was also part of in Trends in Ecology and Evolution, we argue that at least part of these discrepancies are caused by scale. And this idea that there is no reason to believe that trends in diversity are constant across scale. So extinction rates occur at the global scale. Our data comes from the global scale. And there's enough species and enough individuals in this planet to allow to allow enough degrees of freedom to actually have trends going in opposite directions at different scales. So we need to pay much more attention to scale. So that's true of spatial scale, and it's also true of temporal scale. So I'm going to talk a little bit about temporal scale now for just a couple of slides. So this is a plot of the slopes in our data as a function of the start year of the time series. And what I want to show here is two things. One, there really isn't a trend. Older time series don't tend to be negative, and younger time series don't tend to be positive. But also, this is not it's not a random distribution. I don't think our data samples like the past 100 years very well. Like I am reasonably confident that the past 50 years or so we are doing a reasonable job, but certainly not any time beyond that. In contrast, the predicts paper is about what's happening in the planet in the past 500 years, and really I don't think our data is directly comparable with that. It's just like we're talking about different things, and again, the same argument that applies to the spatial scale, I think, applies to the temporal scale. And that's even more true of the mass extinction, because we really require current extinction rates to stay this high for 20,000 years for this loss of 75% of species to have occurred. What we have, what our data has to say about this is, very little. So I guess to keep with the fruit analogies here, I think part of this apparent discrepancies are really because we're comparing apples and oranges. Okay. So, so far I've been talking about alpha diversity. So this is another way of looking at our results. We have R square on the y-axis and slope on the x-axis. The red bubbles are the time series where we had a loss of species. The blue bubbles, time series where we have gain of species. And there's lots of gray bubbles that are time series with flat trends. This is the result that got all the attention, and I find that really surprising, because both in the title and every single one of our plots had actually another result, which is a lot stronger. So everyone pays attention to the null result, but we detected a really strong result, which was change in composition. So this is a plot of temporal beta diversity, so how community composition has changed through time, throughout the time series. And the signal that we detect here is pretty much everywhere is changing in species composition, right? And this is true throughout all of our time series. This is what the other plot looks like. And what's interesting as well is that this rate of change in composition is orders of magnitude higher than we predict from two different null models. So this is, this is real, people. This is strong. It's a very strong pattern that we can detect. And surprisingly for me, it seems to be the result that nobody pays attention to in our paper. So just to summarize all this, let me be very explicit about what I think our results show. We found no evidence for systematic biodiversity loss, and emphasis on systematic. And let me explain exactly what I mean by this. I do not mean that we are not losing biodiversity anywhere. We most certainly are. I mean, the predicts work shows that, if you urbanize or turn primary vegetation into a cropland, evidently you lose biodiversity. There's no way around it. And Matt Hansen's talk yesterday, showed effects of deforestation. Again, if you cut down forest, evidently we lose biodiversity. There's no way around it. But that's not the whole story, right? What I think our results show is that we're not losing biodiversity everywhere, and we should also be paying attention to this. And actually, we should probably be using this information to tell us what we are doing right and try to do more of the right things. Okay. What I think our results also change is that there is a systematic biodiversity change. And I think similarly to what's happened to the climate change debate when we started out talking about global warming, and we now, like everyone talks about climate change instead, we need to do the same thing for biodiversity. We need to have a nuanced and more accurate representation of what's going on in the planet. And what I think is definitely happening is a global reorganization of species in space. And there may be some pretty self-evident culprits for this. So, and one of these drivers, I believe, is climate change. I mean, there's also species invasions, but I just want to give this example as one that I think is probably particularly important. And so I want to highlight a work by Garcia Molinos and colleagues that came out last year in Nature Climate Change, where they projected climate change and then they projected change in species richness as a function of climate change. And what they found is that actually there's a bunch of places in the world that we expect to be gaining species because you have species migrating from the tropics towards the poles. Right? So some parts of the world we expect to be losing species, and some parts of the world we expect to be gaining species. So we need to be paying more attention to that. And again, this is a completely independent line of evidence that supports this idea of a global reorganization of species in space. At a very different spatial scale, so work that Anne led and that came out in Nature Communications last year, we looked at species assemblages in Scottish seas, and specifically we looked at both alpha diversity, species richness, but as well as spatial beta diversity, and we asked how has it been changing in the past 30 years or so. What we found is that actually communities are becoming more and more similar to each other through time. So spatial beta diversity is going down pretty consistently across these assemblages. And what is actually, like, if we go and deconstruct these results, what we found is that this is happening because the northernmost communities are becoming more similar to the southern communities. And again, a signal that species are migrating polewards, and entirely consistent with the work of Garcia Molinos and colleagues. So again, evidence for this global reorganization of species. Okay, so but enough about communities. Oh my gosh, am I running out of time already? We can also use assemblage time series to look at population trends. And this is a plot of population trends for species that have gone extinct within our time series. We focus on time series with 10 years or more here. And as maybe we would expect, species that are going extinct, they tend to have declining population trends. But what's kind of interesting is that this is a very tiny fraction of the species in our assemblages. And there's a comparable or even higher number of species that are actually colonizing and increasing in abundance. And then there's the overwhelming majority of species that are, some have declining, some have increasing trends, some have very stable trends, and they're sort of maybe flicking in and out, but then neither going extinct nor colonizing. So. And the point that I'm trying to make here is that to understand biodiversity change, we can't just focus on extinction. We need to look at the whole picture. And if you were at Melody's talk a couple of days ago, you will hopefully be aware that the importance of invasions and aliens in contributing to biodiversity as well. Okay, so I hope I have convinced you that assemblage time series are a very powerful way to sample biodiversity. We can get 3 out of the 6 classes of essential biodiversity variables from this sort of data. We can get population trends, we can get composition, and coupled with trait data, we can get changing traits as well. And I think Amanda was talking yesterday that, for example, we can get CTI out of these sorts of data. Towards the future, so this is the spatial distribution of the data that we assembled for the science paper. We haven't stopped there, and actually, I believe the ERC announced yesterday Biotime's been funded for a little bit longer. So we're going to keep growing this resource and hopefully make it more. Make it available and more useful. Since the science paper, we have nearly doubled the number of records in our database. We now have 11 million records, over 600,000 locations, over 38,000 species. We've also gotten a lot better at cleaning these data. So we lost a bunch of species when we did this. So just be aware. And we've also combined our efforts with Mark Vellens and Robin Elahis. And more data are welcome. So if you have data, if you know of data, do get in touch. OK, so actually, before I move on, one thing I want to point out. So we've nearly doubled our the size of our database. But if you compare what we had in 2014 with what we have now, you can barely tell the difference. And this map should be kind of familiar by now, because it's not very different from the predicts map in terms of the gaps. It's not very different from the LPI map in terms of the gaps. So if we go back to this idea, of wanting to get global stratified random sampling, it becomes very clear that this is not enough. Like, no, just assembling existing data is not gonna, we can do better, and we are doing better, and we are specifically targeting places that are empty at the moment. In fact, last night I got someone from Russia getting in touch, which is great, because we don't have a lot of data from Russia at the moment. But frankly, we need to do better. If we're serious about approximating randomized global sampling of the planet, we need to do better. What's important is that gaps are not random, right? They're not there because we've been distracted. These gaps are there because they are difficult to sample. They correspond to remote parts of the planet, places where we don't have experts, or we don't have a lot of people, or are just like really difficult to sample. They are also overwhelmingly tropical, which is a problem, because we actually think that climate change makes the tropics particularly vulnerable to loss of biodiversity. And it's also a problem because our sampling designs are not really well geared for sampling the tropics, right? If you sample, if you sample tens of individuals, that's okay if you have 30 species or so, but it's not okay if you have 3,000. So we can't expect the same sampling protocols to work in the tropics as they do elsewhere. So the bottom line is that what's left to do is difficult. And what I think this means is that just more of the same is not, it will not work. We can't seriously approximate global randomized sampling of biodiversity on the planet, but just keep doing what we're doing. We need to sort of think outside of the box and do better. And so what I want to point out here is that in the same way as we no longer use these things, floppy disks, which some people might call, I've heard someone say this, that, someone 3D printed a save button. No, so we don't use these things anymore, so we should also not be using necessarily the same technologies now to sample biodiversity that we've been using for centuries. We should harness technology in our bid to do better in biodiversity sampling. So what do I mean by this? I think we need to be much more efficient at sampling biodiversity, and this will, I don't think this can be done if we don't move to some extent to automated sampling. I mean, we don't, like, weather stations do not have mercury thermometers where someone goes in with a notebook and writes down the number in there. So we can't really expect to do the sort of sampling that I think is needed using the same sorts of technologies. We need to move towards automated sampling. We also need to be much better at distributing our sampling effort. And running the risk of saying something incredibly unpopular, I think that means that we need to really target our investments in biodiversity sampling away from Europe and the US and Australia. And I'm sorry to say this, but those should not be our priorities. We need to tackle the deep sea, the biggest habitats on our planet, and tops of mountains and the tropics. So these are the places that we have not. These are the gaps that we have. This is what we really need to do. We also need to move towards multi-scale data sets. I mean, like I said, I think overwhelmingly we think that scale matters, and the only way we can really tackle scale is by collecting data that explicitly allows us to quantify the effect of scale on biodiversity patterns. And I think we need to think smartly so that we collect data that allows us to quantify as many essential biodiversity variables as possible, ideally all of them in one go. And also that allows us to link the different essential biodiversity variables. And finally, I think we need data that allows us to parameterize forecasting biodiversity models. So not just any data, not just data that we can collect, data that we need to collect. So we need to get theory and data collection much more well coordinated. Okay, so since the S-Change working group, Martin and I have been talking about this idea that we kind of need, it's time for a Space Hubble telescope or a Large Hadron Collider for biodiversity sampling. And I've been kind of playing in this idea, what would that look like for biodiversity? I don't think it's a single instrument. I think it's probably a constellation of stuff. It's probably a constellation of dedicated satellites that do the global coverage, large-scale stuff. And then it's a fleet of robots, drones and underwater drones, that go around photographing everything and building really detailed imagery for that we can't get from the satellites. This is an example of a robot that Steph Williams at the University of Sydney owns, and we've been playing with it on coral reefs. And I was at the MBON presentations, and there's amazing work being done with eDNA, and I was thinking, if we can put an eDNA sampler on one of these things, we can do both. And by then, they will be able to kind of link satellite images and aerial images and underwater photo mosaics annotated by people. And then we can use these annotations, and we can actually automate the identification process. And that means that we will be able to do all 6 essential biodiversity variables in one go. So this is my vision for the future of global biodiversity sampling. Thank you.
Thanks, Maria, for such an inspiring talk, and we have time for a few questions.
Great talk, Maria. Very inspiring. Are there any major taxonomic gaps you've noticed in these time series, like how well are insects represented and the most diverse taxa?
There's definitely gaps, and that's the other thing. Well, I kind of stayed away from that because it's another hobby horse of mine. Invertebrates are always grossly underrepresented relative to their contribution to biodiversity. Yeah, so that's the other thing that we need to do. We need to be part of the stratified sampling, I think, has to be about taxon as well. And we can't just go with what's easy. We need to go with what we think is important. And that's where I think we probably need to talk to the ecosystem people and ask them, what are the things that really matter for ecosystem function, if that's one of the drivers of understanding biodiversity change. That is one of the ways in which we need to do better.
Hello. I just wanted to ask if you disaggregated either the species richness time series or the population time series by protection, like protected areas versus non-protected areas, and then whether there was a difference.
We haven't yet, but there is ongoing work. So one of the things that we're doing, so I guess when we started doing this, I felt that we didn't quite have enough data to tackle that question well. It's partly why we wanted to grow our database. And I know it may sound a bit crazy, like, what do you mean you have 11 million records is not enough data, but it's not when the universe of what we're trying to sample is so large. It's partly what we want to do in this phase 2 and it's part of the work that we think will come out of this S-Change working group is looking at that and looking at different, not just protected areas versus non-protected areas, but different governance approaches and those as drivers of change.
Thanks, Maria. So if you're advocating for new sampling in the difficult places to get to, do you have a sense of the sampling interval, the minimum sampling interval to get some measure of change after the first sample?
That's a tough one. And I think it's probably system specific. I think, for trees in forests, we can get away with not going back for numbers of years because their dynamics are quite slow. For assemblages that with faster dynamics, we probably want to have a minimum of yearly sampling. So currently, although we have more detailed information for at least some of our time series, we're using year as our minimum grain, temporal grain, and that's partly because if you go below that, the seasonal effects make the whole problem more complicated. So I don't know. This is something we should ask the data actually. Is what is a good sampling interval? And I suspect that's probably going to be system specific.
Marie, thank you very much. This was a great talk. I had a very similar question, and I was wondering if we could pinpoint you a little bit on the idea of how do you separate the idea of random sampling and stratified with time series, and are there any specific areas that you think that are priority areas we define areas to go into with an MBON?
Well, I think we need to, well, I haven't thought about that carefully enough to give you a good answer, but actually from Matt Hansen's talk yesterday, I was thinking that we should probably be using large-scale information about what's going on in the planet to help us target this run. Well, I think we need a combination of things. So we need consistent monitoring that always happens regardless of what's changing. And then we probably also need to have sort of targeted sampling when we think there's something major going on. But again, I think we should have a responsive mode of that's not the right word for it, like an adaptable sampling protocol that is probably two-pronged. Does that answer your question or not really?
Marie, the comments you've made on the relationship between change and homogenization and the effects on what you see in alpha and beta diversity because of that, do you see a challenge more broadly in us communicating this clearly, and do you have ideas on how to get the concepts across so that they're not buried in the big picture stories from other scales?
So are you asking me if I think we can communicate change in biodiversity, or are we stuck with species richness?
No, I'm talking about communicating. I mean, you started with the point that there was an apparent paradox that would appear to many people about the stories of global extinction and the messages coming out of some of the data sets being focused purely on the alpha diversity slopes. Is this a communication challenge we've got to overcome, or?
No, I think it's a habit. I think we can easily communicate change in composition the same way as we communicate change in species richness and extinction. I don't think that's any harder to communicate than one than the other. And people can probably relate to this. We can talk to them about the birds that they see in their gardens and how they have changed through time or not changed through time, So I think it's a habit more than something that is impossible to communicate. And I don't think, the general public can get excited about Higgs boson, right? They can totally get excited about beta diversity, which is way more tangible and closer to their daily lives. So.
Okay, maybe you'll still be around for answering some more questions. Again, thank you for this excellent presentation. Really